The title of my talk today is Now is the Time for Molecular Therapy for Angelman Syndrome. So that's the subject that I want to focus on is why is now the time? Why is this a good time for molecular therapy for Angelman Syndrome? So let me start out first by saying, what is molecular therapy? So molecular therapy we usually differentiate from drug therapy. And drug therapy, which is actually still molecules, but drugs are something that you would have to take over the course of a lifetime. Usually the drugs don't cure people of something, but they're reducing the effect of whatever is the problem, and so you can take these drugs and be better. We talked about drugs yesterday, and we're all about drugs. If we can find drugs that will help in Angelman syndrome, we're going to find those drugs through our infrastructure program and help promote that process. So there's nothing against drugs, but when we talk about molecular therapy, we're usually talking about things that are happening on these other levels, the level of the RNA and the level of the DNA. And so in Angelman syndrome, we have a DNA problem, right? We have a deletion or a mutation that's happening on the DNA. And so if we could treat this disorder on the level of DNA, that's really getting to the root cause of the problem. It's not something that this problem has caused, and now we're treating the symptoms of this. We're going right to the root. What is the problem? It's a genetic problem. Can we fix the genetics or address the genetics? So that would include things like gene therapy, gene editing, epigenetic editing. Some of these terms we might have talked about yesterday. Things like shRNA and microRNA, those are working on the RNA level. So that's one level up from the DNA. And things like antisense oligos are also working on that RNA level. So we can imagine that in our cells, there's usually just 2 copies of every gene. And so if we can work on that level, we only have 1 or 2 molecules to worry about, to try to fix. That DNA can make many copies of RNA, so now we have more copies of RNA that we would need to address with an shRNA or an antisense oligo. But those therapies still work. And then, of course, those could make many proteins, and so when we get to the protein level, there's a whole lot of molecules. But we're going to be talking about things on those top 2 levels today. So, number 1 reason. What is the number 1 reason why we should be excited about molecular therapy for Angelman syndrome? And the number 1 reason is that cell and gene therapy is now a thing. So this is really profound. This is a chart showing some of the gene therapies that have been now approved. Starting with Glybera over here. It's the first one was the first gene therapy that was approved, that was actually approved in the UK in 2012. And then Luxturna was the first gene therapy approved in the United States in 2017, so not very long ago. And then you can see there are a few more. Not everything is on this slide, but it gives you the idea that all of this has happened very recently. So in my career, for example, this is an amazing thing. Most of my career was looking at gene therapy like this, where in the history of gene therapy, there were some successes and failures and successes and failures. And we were always talking about what gene therapy was going to be like, when it was really happening. And, I would have classes and I would tell my students, tell us about the progress of gene therapy. Tell us, which of these gene therapies look like they're going to be useful. And finally, in 2017 and 2012, we could point to some gene therapies that were actually approved for use as medical products. And so finally, we were able to get to a stage where there actually are gene therapies approved. And this is really important for all these molecular therapies that we're talking about, because I just want to impress upon you that this is a thing that's happening. This is not something in the future. We're in this gene therapy age now. So this is just one of many slides that I could show talking about gene therapy. And so in this update from 2020, there were about 6 diseases that were being treated with gene therapy. There were like 360 that are in the pipeline. So there's a lot of even that we're just seeing the tip of the iceberg now with those things that I showed on the slide. There's lots and lots of gene therapies now in the FDA approval process, and a lot more that are under development. So, what we're doing in Angelman today is we're riding a wave of being able to use these technologies to treat disease, and this is a real thing that's happening now. So this is not futuristic. This is now, but it's in our time. And then following on the heels of gene therapy, where we're trying to take a gene and just put it into the cell, we have things like gene editing, where we can not just put a new gene in the cell, but we have the opportunity to actually change the DNA in the living cell. And this comes about with these kinds of advances like CRISPR that you've heard about. And CRISPR can be used in many different ways to do a number of different kinds of jobs. It can mutate DNA in particular places. It can repair DNA in particular places, depending on how those experiments are designed. The very first use of a nuclease or some kind of gene editing tool in a clinical trial was this one that was targeting a protein in the body that HIV needs to infect the cells. And they found that if they could mutate the gene that makes this receptor for the HIV virus, that essentially the virus would just bounce off the cell. And so they were able to do this in the same kinds of cells that Joe Anderson was talking about yesterday, those cells that make all the blood cells. So you could essentially get HIV-resistant T cells out of this. And this is now in clinical trial as a way to prevent people that have HIV from getting AIDS. That was in sometime earlier on. But that was what we might call an ex vivo gene therapy, where they modify those cells in a dish, and then they reintroduce those back into the patient. The first use of a gene editing in a person was in 2017, and this was used to. It's still in clinical trial, right? So not approved yet, but these are to treat some kinds of blood and liver disorders. And in this case, they have to take that CRISPR tool or that gene editing tool, put that into a viral particle, and now introduce that into a person. So this is now in vivo somatic gene editing. And you can imagine that this is a little bit more complicated, right? So we have to go in and into the body. Yeah, we have to deal with the immune system. We have to deal with the fact that there's many, many cells in there. Some of them are packed into organs. Some of them are spread out, like in the blood. So we have to design these systems to accommodate all these differences. The first use of CRISPR in humans was actually a therapy that is being investigated in eyes. And they start in the eyes because, I'll point to this side now. Eyes are a kind of immunoprivileged organism, so we don't have as many immune complications. They work well for gene therapy, and in this case, they also had a mutation that they were targeting in this eye disease that really lent itself to this kind of approach. So again, I'm showing you this to just say that these kinds of things are happening. They're actually being used to do this kind of editing in living cells in people. And with careful design of the setup, they can be safe in people. So this is another example of a use of CRISPR nuclease gene editing, CRISPR gene editing in a person. In this case, it was in the liver, and to treat a rare liver disease. And so you can see that these are just in the beginning of clinical trial. But this is happening, right? So even things like this are moving forward. So when we talk about molecular therapies that we'll be discussing, and that you've heard about at this symposium so far, this is being pushed ahead on all fronts. So we're moving with the wave of the technology for Angelman syndrome. So something that I do feel some concern about in all of this is the prices of some of these things, and I just want to call that out as we're going into the pharma session. So on this axis here, it's actually showing the cost of the therapy for each one of these gene therapies that have come out so far. And so Glybera actually, when it was released, was a $1 million therapy. At that time, it was the most expensive therapy in the history of medicine. The Luxturna one is about $450,000 per eye. So these prices are very high. And one of the more recent ones that was approved, that we heard a little bit about yesterday, Zolgensma, debuted at about $2 million per treatment. And so I think for many of us in this field, we view this with a lot of concern because as researchers, we're often thinking about the technical challenges that are involved in all of this and how we overcome those technical challenges. And there is a concern that these very high prices of these therapies are going to create a real barrier to their use. And I think that we would like to resolve, here today to say that we want to make these therapies as affordable as possible, and we're going to try to ensure that whatever comes out from these molecular therapies, we want them to be able to be used by this community to make Angelman syndrome go away. But that's a thing. So reason number one, cell and gene therapy is a thing. And that's really important because I just, again, want to emphasize that what we're telling you is not going to be like some, maybe in the future when we work this out. It's like this is happening now. So then there's reason number two. So reason number two, why this is the time for molecular therapy for Angelman syndrome. Angelman is actually a good fit for molecular therapy. So for all the bad things that we could talk about with Angelman syndrome and all of the problems that this region causes in the genome and all the weird kinds of genetics that goes on there with the imprinting and the RNAs and the antisense of this and that, the other thing, all that weird stuff that happens, we have to think that there are some good features that make this amendable to this kind of molecular therapy. So for example, when we lose this region as a large deletion, there's still one extra copy. And this is kind of unusual for other kinds of disorders, right? I mean, there's a lot of disorders that when the genes are mutated, your only choice is to do a gene therapy and reintroduce a new gene. But in this case, we have this extra copy of this gene that's sitting on the chromosome that is silenced, but if we could unsilence it, we would get a gene, in principle, that should be in the right physiological location. It should be under the natural regulation that gene would have. And so the natural regulation might be an important thing. There is some evidence that the activity of the gene that makes UBE3A might be dependent on whether those neurons are getting stimulated at that time. There's evidence for that. So some of this can be recapitulated with gene transfer therapy, but if we can actually activate that gene that's right there for us, that gives us a special opportunity. And this is not an opportunity that is available for most genetic diseases. So, I mean, can we say that with all of the unlucky things that happen here, I think we're fortunate to have that kind of situation where we could design a therapy to take advantage of this kind of Achilles heel that Angelman syndrome has. And then it does seem like if we could repair this one gene, we might be able to take care of a lot of the symptoms of Angelman syndrome. And that's also a good thing too. So, lots of other diseases like cancers, like autism, you can't point to one gene and say, if I could just fix this one gene, then all this disease would go away. But in the case of Angelman syndrome, we really feel like that could be possible. So for these kinds of reasons, it does seem possible that we could design some molecular therapies for Angelman syndrome. So you've seen a lot of this kind of thing before, but I just want to put it all on one slide, because when I see it all on one slide, it's just really overwhelming, all of these shots on goals like we talked about yesterday. So for sure, you could come in with some kind of a adeno-associated virus, just deliver a UB3A gene into the cells, and maybe that does everything, right? Maybe that takes care of everything. We'll have to see. But there's people that are working on this. I think there's at least 3 companies that are proposing to do gene therapy for Angelman syndrome. And we have to see how this is going to pan out. But from the Translational Research Symposium that we heard on Thursday, it seems like this is really moving along. So we're hoping to see some clinical trials start hopefully soon. That'll be really exciting when that happens, to see a gene therapy. Then, of course, there's that kind of therapy that Joe Anderson talked about, somewhat similar, right? So there he has a virus that he puts into a different cell. That different cell gets into the brain and puts that UB3A protein right into the neurons. And so you could see from yesterday that that was really an exciting approach that seems like it might be working too. So we're looking forward to seeing that in clinical trials too. So that's also something that's coming up. Then there's antisense oligonucleotides. So that's working to chop off the end of this little blue RNA. And as we know, antisense oligonucleotides have many advantages in this molecular field. They're relatively easy to make. They're relatively cheap to produce. And for those kinds of reasons, it makes total sense that those were the first to go into clinical trial. And even though that makes sense that they go first into clinical trial, it still took a very big push to get them into clinical trial. And we should all be proud of FAST. I can say that as I'm not part of FAST. I'm associated with FAST, but as a scientist, it's just been amazing to be part of this group, to watch this happen, to see this idea of these ASOs go from conception through lots of different trials or designs and experiments to actually being the very first clinical trial of a molecular therapy for Angelman syndrome. So that's really exciting. I think the whole community should be proud of that. Yes. Yeah, thank you. And so the ASO is doing this job of stopping the stop. And so this UB3A gene now is free to express at whatever level it needs to be. It shouldn't be too high, it shouldn't be too low. It should be whatever the body intends it to be. And then there's an approach where you come in with an AAV and you could add a Cas9 to cut the DNA, or a Cas13D to cut the RNA, or something like a short hairpin RNA or a microRNA. And we heard about all those kinds of approaches yesterday, I think. And so, when you look at all these different ways that we're trying to do molecular therapy, and these are all based on the special genetics that happens at this locus. So we're using this situation that is causing the bad thing, but we try to find that Achilles heel and design a therapy with these modern tools that I've just told you are being used by many people to try to do therapy today. We're going to put those same kinds of tools to work in the case of Angelman syndrome. And who would it help? There was a lot of talk about this yesterday too. It's going to help everybody. So certainly in the case of deletion, when there's not UB3A being made, if we could reactivate the silenced copy on this chromosome, that would be helpful. In the case of UPD, I guess there's a concern that you might have double activation and double the level, but we'll have to see. It might not be, I think Allison showed a slide maybe on Thursday talking about the level of UB3A activation that has to happen to rescue symptoms of Angelman syndrome. And it doesn't have to be at the roof, right? If we raise the levels to a sufficient level that we could overcome the problems caused by loss of UB3A, that might be enough. And that's probably below the threshold that starts to cause problems with Dup15q. Imprinting disorder, same kind of thing. These same molecular therapies that we're talking about will turn on UB3A here. And even with point mutations, that we're not going to fix that mutated copy, but we'll turn on that silenced copy, and that will provide the UB3A in the brain. So it should be a therapy that should work for all cases. So we're going to push forward with that and make sure that that's going to happen. So reason number 1, cell therapy is now a thing. Reason number 2, the genetics is a good fit for molecular therapy. And reason number 3 is you. So I want to say that FAST has really done a lot to change the course of this disease. And from the very beginning, they took a very aggressive approach to raising funding. And we're seeing the results of that aggressive approach now with all of these different molecular therapies that are now in the pipeline. As Allison likes to say, that there's now 27 different companies that are coming in. 28, sorry, I didn't mean to leave that last one out. 28 companies that have come in to work on Angelman syndrome. And that's an amazing thing. I mean, now there's like a buzz about Angelman syndrome, right? So that's... And the fact that that not only did they raise this money, but also be able to have a company then take this to the FDA and just have everything lined up. Not just even the molecular therapies and what happens after that, but also the ABOM, for example, which is this group that is trying to look at clinical outcome measures and organize all the efforts around that, because that's a critical component for the success of any therapy when it goes into clinical trial, is what are those outcome measures and how can you know when this is really working in a way that's going to work. And I could just say, I've worked with other kinds of foundations, and some of those foundations, they try hard too. They have good organizers too, but the community has just not been working out as well. There's less people showing up for the meetings. There's less fundraising that they're able to raise. They're not as able to attract in companies. They're not as able to attract in researchers. And it's hard. It's hard out there. For many diseases, rare diseases that are trying to find someone to help them with their situation, it's not easy. And sitting here with this community, it just strikes me how important it is to have a community like this to support the efforts of FAST, to support the researchers, to support all these efforts that are going on. So Allison said that I should tell a little bit about how I got involved in this as well. So I actually was just minding my business, and I got a phone call one day from a Google group. But this is before Facebook. There was no Facebook back then. And there was just a Google group of concerned parents and clinicians. And they contacted me, and they said that they had read some of my papers, and they were interested to know if the technology that I was working with could be useful for the treatment of Angelman syndrome. And I have to say that I'd never heard of Angelman syndrome back then. And so I looked it up on that vast repository of human information, Wikipedia. And that was the first time I actually learned about Angelman syndrome. And then, just from that, I could kind of understand what it was that they wanted to do. They wanted to reactivate that gene. And so I wrote back to them and I said, well, I think we could actually design something to do that, but I'm not sure, if doing a late stage intervention is going to be helpful. And so they said, oh, he still thinks it's a neurodegenerative disease. We have to send him this paper. So they sent me a paper by this guy named Ed Weber. And that was the first time that I've heard of Ed Weber. And eventually I met with Ed Weber and at a golf course for an event that Mady Dunnigan had put together. And so I actually went to meet this Ed Weber guy, and so he was out golfing, and we ended up talking to the golfers about like ubiquitin and neurologic activity, and that was an experience. Those were the good old days, huh? But there was another woman there that I kind of recognized from that Google group, and but I wasn't sure exactly what she was doing, and she said, "Yeah, my name is Paula Evans," and that was when I met Paula too for the first time. Still gives me chills. But I didn't know at that time that that Angelman syndrome would go on to be a major part of everything that I do in a professional way. So I think the take-home message that I want to leave you with is that as parents of children with special needs, we appreciate that it's a difficult life, and you probably feel like this thing has got you, and there's not much that you can do. But there is a lot that you can do, and this is my point. It's just that you should recognize the powers that you do have. And I think that talking to scientists and trying to get people involved, trying to build that community, trying to reaching out and just making this community, building this community, and making everything that we've put together over these many years. This has all come from all of you. And so I just want to thank you for everything that you have done and that you will do, and that you should continue to realize that this community does have the power to alter the course of this disease. And that's why we're going to win. Thank you.